Hi everyone, this is Shiva again from We Talk Science. This time we're with video, so it's not just a podcast. I'm here with Jeremy Barr, Dr. Jeremy Barr from Monash University. Hi Jeremy, thank you so much for joining us today.
No, thank you, Shiva. Great to be here.
Excellent. So you work with a lot with bacteriophages. And before we go into that subject, I would like you to introduce yourself, please, and maybe you can tell us a little bit about your history of how you got into this field.
Yeah, absolutely. So my name's Jeremy Barr, and I'm a senior lecturer at Monash University in the School of Biological Sciences. And how I got into phages, maybe a short abridged version, is kind of a cool story. So I was doing my PhD in microbial ecology, actually, and I was running these really large bioreactors, and we were studying how specific bacteria were breaking down waste nutrients. And a phage got into this reactor, and it actually killed it and wiped it out, and caused it to crash and completely fail. And so I thought it was the worst PhD ever, but we ended up investigating it a bit more, and we found out a phage attack caused this crash, and we put out a pretty cool study looking at how a phage infection crashed that reactor. And then after that, I ended up going and doing my postdoc with Forrest Rower over in San Diego State, and I was with him for 5 years, where I really learned how to be a phage biologist. And since then, started my own group at Monash University about 4.5 years ago now.
So let's go back to the basics. What is a bacteriophage?
Yeah, so a bacteriophage is really simply, it's a virus, but it's a specialized virus that only infects and kills bacteria. So these phages, bacteriophages or phages for short, are highly specific, and they'll recognize their bacterial host, which they'll infect, and they can then replicate, lyse them, kill them, and propagate more phages back into the environment.
Okay, so how are bacteriophages different from other viruses? I mean, of course, for example, the coronavirus. What is the difference there?
Yeah, it's a great question. From a purely biological point of view, they're very similar, right? They are a virus. They have a capsid or outer structure very similar to COVID or many of these eukaryotic viruses. But these phages, I guess, the key difference is that they're specialized in recognizing and infecting only their bacterial hosts. And so it's a really common question that we get, right? So would a phage ever infect a human being and cause a disease? And the really simple and short answer is that no, it won't, right? It'd be akin to an elephant, magically evolving into a beetle overnight. It's so different to, a eukaryotic virus, and it's so specialized to bacterial hosts that they really will only recognize and target bacterial hosts for their infection cycle.
That's really interesting. So, can we go into the biology of this just a little bit? How does a phage recognize its host? So, you basically, if you what you said before is that a particular phage will only infect and invade a particular bacteria. How does that recognition actually happen?
Yeah. So phages are really amazing from my point of view, and they're just so biologically, they're so unique and different. So I'm not sure if everyone's seen a phage, but if you haven't, you should definitely Google bacteriophage, and the first thing that'll pop up are these really alien looking structures, right? They've got this sort of icosahedral head and then this long specialized tail structure. And this is probably one of the differences that we see to human and eukaryotic viruses, is this presence of this specialized structure. And this tail structure, this tail fiber, is really designed and evolved over time to recognize a bacterial host and being able to penetrate across that bacterial host's cell wall and cell membrane. So, some of the specifics is that phage will use that structure to recognize different structures in a bacterial host cell. That might be any sort of capsule or some sort of macromolecule that's displayed on the cell, or a outer membrane transporter, or a flagella. And a phage will recognize that structure, and it'll absorb or bind to that structure, and it will then go through this sort of complex mechanical transition where it'll use that tail fiber to penetrate the outer membrane. It will degrade a small part of the bacterial host cell wall, and will eventually eject its genome inside the bacterial host cell.
Okay, yeah. So, I mean, I can imagine that a bacterial surface, when a phage actually encounters the surface of a bacteria, that's a very different sort of surface than, for example, a mammalian cell. There is this, obvious presence of a cell wall in some of these bacteria. And so, do you reckon that the phages have somehow evolved to sort of recognize these walls and these, extra sort of layers that a bacterial cell has versus a mammalian cell, which is literally just a lipid membrane?
Absolutely. The huge evolutionary history of phages has allowed them to recognize and absorb and bind specific, outer cellular, outer wall structures on these bacterial hosts. And this has really been specialized to the nth degree, and it's something that we see quite commonly in phage biology, is that phages will target a bacterium, but they're actually much more specific than that. And when you'll have a phage, it might only target a specific bacterial species,
Yeah.
such as an E.
Hmm.
coli bacterium or a Bacteroides bacterium. But there's even another level of specificity where those phages will only target specific strains within that bacterial species. And this has been this long sort of evolutionary back and forth between bacterium and phage, where neither's one, and both are constantly replicating and predating on the other. And it's caused the phages to specify and really target specific structures to infect their bacterial host. And it all comes down to that sort of mechanistic recognizing those outer cell wall, outer membrane structures that allows the phage to infect the host.
That's really interesting. I did not know that. So, what the ultimate goal of a bacteriophage would be to lyse the bacteria, its host. Is that correct?
That that is correct, but I would take it a step further and say the ultimate goal of the phage is really to replicate and propagate itself.
Of course.
And to that, there's actually multiple different phage life cycles. And so the most common one that we talk about is what we call the lytic phage life cycle, where the phage will recognize the bacteria, it will infect the bacteria, inject its genome, and it will replicate inside the bacterial host cell using the host cell machinery to produce new phage genomes, new phage structural proteins, which will then assemble into phage particles. And eventually it will lyse that cell from within and will release tens to hundreds of new phage particles back into the surrounding environment. We call that the lytic life cycle. However, there's alternate life cycles, and one of the other major alternate life cycles is the lysogenic life cycle. And here, instead of killing the bacterial host, the phage will infect, and it will inject its DNA genome, which will then integrate inside the bacterial host chromosome, and it'll lay dormant in what we know as a prophage. And so in this life cycle, the phage isn't necessarily killing the bacterial host cell. It's really hitching a ride, and it's persisting alongside. And whenever the bacterial host cell divides, it will also divide and replicate that prophage. That prophage can come back out into the lytic cycle, but it's a unique example where the phage and the bacterial host cell are almost in a symbiosis.
So that I reckon that whether it goes into lytic life cycle or if it basically becomes a prophage depends on the environment in which it is. I'm guessing that this is like my guess, that if the bacteria is in a environment that is not so conducive to replication and is not as happy, then the phage would go into prophage, waiting for better conditions, so to speak, I suppose.
That's exactly right. And there's lots of really exciting research, in this space under what sort of different environmental conditions or condition of the bacterial host cell does the phage choose to, replicate and produce more progeny via the lytic life cycle, or integrate and persist and reside within the bacterial host cell. And it really gives, from the phage's perspective, it really gives it a whole new array of different lifestyles and potential ways to propagate itself and maintain further.
Wonderful. All right, so let's talk about a little bit about what you do in terms of bacteriophage. I understand that you're working on the virome, so essentially the phages that exist within us. Can you tell us a little bit more about that, please?
Yeah, so I would say that the work that our lab does at Monash is, holistically we are looking at the interaction between phages, their bacterial hosts, but also the interaction of both those entities with the mammalian or the human host. So something we call a tripartite symbiosis, where phage, bacterium, and mammalian host or eukaryotic host are all interacting and interplaying with each other. And the best example, or the best ecosystem, in my opinion, where this happens is the human gut. The human gut is our major organ for digestion, but it's also a huge microbial incubator. Each of us houses trillions and trillions of bacterial species that reside within your gut, and these are hugely important for digestion, for your immune system, for your day-to-day function. There's even suggestions that these gut microbes control the way that you think and the way that you feel, which is perhaps terrifying or exciting, depending on how you look at that. But behind all of this sort of microbial interplay are the bacteriophages, and the phages in your gut microbiome, are incredibly complex, incredibly diverse, amazing, and we've barely scratched the surface. And so our lab is looking at this from a range of different approaches. We're looking at it from a discovery approach, figuring out, who are these phages, how abundant are they, where are they present, through to more mechanistic, how do they work, how do they function, how do they control the bacterial host, how do they influence that microbiome ecosystem.
Right. Okay, so tell me more about this, about the phages that exists within the gut of the human, I guess, microbiota. What are they doing there? I mean, I understand that where there is bacteria, I'm sure that there are phages, but what is the function of phages that are sitting inside your gut?
Yeah, it's I would say it's still an open-ended question. There's a lot of work going on here, and I'll maybe give two perspectives to sort of answer that. And if we look at the pure lytic perspective, so we know that the phage will infect a bacterium, and it'll propagate and it will kill that bacterium, and that happens in the gut. And so from that viewpoint, these phages are controlling the bacterial species and diversity present within your gut microbiome. And so you could look at this from a range of different ways. Say if you had a pathogen come into your gut, might cause food poisoning, might cause you to get sick. Theoretically, you could apply a phage, or there might be some phages present that could attack and kill that microbe and keep that under control. But this is happening on a grand, innumerable scale, where we have a huge diversity of bacteria and an even huger diversity of phages. And so it becomes this really complex ecological environment where no single bacterium will ever really come to dominate that system, because as soon as they do, a phage will pop up that will knock it down. And so the phages really behave as this sort of top-down predator that really suppresses any one bacteria from dominating, and keeping a healthy gut microbial ecology. And then you can look at it from the other point of view, being the lysogenic life cycle, where the phages will integrate and persist and live as part of the bacterium. And this is, from recent research, seems to be the more prevalent and dominant life cycle that we see within the gut. And this has a lot of really interesting implications. We know that phages encode a whole range of different genetic elements and proteins and enzymes in their genome. And when they integrate into a bacterial host chromosome, they transmit that information. And so you have this potential for something known as lysogenic conversion, where a phage can gift its bacterial host a new function that it didn't previously have. And in the context of the gut, we're looking at how these phages may contribute new metabolism, potentially new virulence factors, or new ways that those microbial hosts may persist in the gut, establish, and how they contribute to your own function in your microbiome and your health.
Okay. So how do these new phages get into your system in the first place? So you just mentioned that they introduce new genetic elements and to change the what's happening within your gut. Where do these phages come from exactly?
Yeah, another really, really interesting question. And the I'll maybe approach it and say that there is constant input into your system, right? Every time we eat food, we ingest something, you're going to have a new microbe potentially coming in. But we don't tend to really see that as a major influence. And one of the amazing things that's come out of gut microbiome research over the last really 5 to 10 years is that the adult gut virome is quite stable. And so what researchers have done is they've looked at the virome from an adult human over 1 to 2 years, and they've seen that although it is really diverse, that virome is quite stable over a number of years. But when we compare between individuals, that doesn't seem to be the case. So two individuals will have a completely unique virome, but each of their unique virome sort of persists and stays with them throughout their lifespan. And so it seems to be that as you approach adulthood, you've sort of gone through this tumultuous period where you've got new microbes and new viruses coming in. But as you approach adulthood, that somewhat settles down, and you harbor a virome that appears to persist with you throughout your adult life. And so what researchers are now looking at is how this virome initially is seeded. And so what we're starting to begin to understand is that during childbirth and breastfeeding, and as the infant's weaned, a lot of influxing bacteria and viruses are fed into that infant, and that's really the starting point for their microbiome and how that persists and eventually matures into adulthood.
Okay, so that's really interesting that the virome essentially persists and remains fairly stable over the lifespan. We spoke about, of course, earlier about how researchers, of course, are also looking into perturbing that a little bit or introducing new genetic elements via phages into the gut microbiome. For my first question is this possible? Like, has this actually already been done? And my second question is, should it be done? Because if the virome is stable throughout the lifespan of a human being, maybe perturbing it is going to actually cause more damage than benefit.
Yep, absolutely. So coming to the first question, has it been done? And it has been done to some respects, or it's at least tried to be done. And so there's been a lot of studies and examples where researchers have tried to apply a phage to a gut microbiome, and that could be to perhaps clear a bacterial infection. So you might have a patient or an animal model that's been colonized by a specific gut bacterial pathogen, and by applying a phage to that system, we can hope to knock that down or eradicate that. That hasn't really seemed to be the case. So what we've seen is that some of these bacterial pathogens persist at a really low level, and although they cause disease and make you really sick, phages don't really seem to eradicate them. Rather, phages are more a balancing pressure. And so what's sort of what we're seeing, how we're seeing phages behave in the system is more of an ecological sort of pressure to level that bacterial playing field and increase the diversity. Some other aspects people have applied phages is in the sort of field of fecal microbiota transplant. So where you have a patient colonized by a single bacterium that might be antibiotic resistant, and by transplanting a microbiota and also transplanting a phage, you can restore some of that diversity. It kind of comes to this question. So your second question, should it be done? It's an interesting question. I believe that when we understand this system, which is probably still a whiles off, that it will be done. And I think that we'll almost be tweaking and modulating this system in a really fine way, fine scale, that we can increase someone's diversity or decrease it. And I think phages will be a really great way to do that. And I'll maybe add on one more question to that, or one more point to that, is that how is that going to be done? Because the gut is such a complex system that it's really difficult to study and understand. So something that we're doing in our lab is actually building a lab on chip, so building a small device that we can recapitulate this gut environment, and we can actually empirically test this. We can add this phage, we can add this bacterial host, and understand how these interactions occur. And with that understanding, hopefully we can one day manipulate the actual human gut.
Wonderful. Yes, of course, it's the organ on a chip, or the gut on a chip, or an organoid on a chip. I mean, I think everybody's, really, it's such a hot topic these days. That sounds amazing. The other question that I just that I had was, there's been a lot of studies that have been published in terms of the microbiota and the microbiome changing with different conditions. So there is correlated studies with diabetes, obesity, all of these different conditions, IBD, for example. What is, if any, do we know what the phage or the virome actually plays? What role it plays in these sort of conditions? When things kind of are not normal. What's the role there?
Yeah, so I would say we're not quite there yet, and I would say that we don't quite understand what the virome is doing. And I should maybe just clarify one point, that when I'm talking about the virome, I'm specifically talking about the bacteriophages. There are other eukaryotic viruses, but for the most part, your virome's dominated up to 95% by bacteriophages. And so we don't really know what the virome is doing. We don't really know what the virome's doing in healthy people, let alone what it's doing in some of these people that have some of these diseases. But what we do know is that there are differences. So I'd say where the field is at right now is completing some of these correlation studies. So we know, for instance, that in some of these patients that are suffering some of these inflammatory diseases, Crohn's or IBD, that they appear to have a decreased viral diversity, yet increased abundance. So what that means is that there appears to be more viruses, but there's less diversity, or there's high numbers of a single type of viruses. And this, now, one caveat to this is this may simply be a reflection of the bacterial diversity. A lot of these patients also tend to have a single dominant bacterial phenotype. And it kind of comes back to the question, well, is the virome really shaping that microbial gut ecology, or is it really mirroring it? And this is still an ongoing argument in the field.
I think this is also part of the broader question on the microbiome field in general, right? Like, what is the causative agent, whether it's the bacteria or the ohm or the biota that is causing the phenotype, or the phenotype causing the differences in the microbiota. And I remember having this question years ago when I first attended my first microbiota conference, and I guess people are working towards it now in terms of mechanism and things like this, but it's still such an open-ended question.
Yeah, and I think it's just such a complex system, right? And the reality is it's somewhere in the middle, right? It's the virum's going to be both correlative and causative to some degree, and it's what we're really seeing is that every person's gut is unique. Every person's gut is their own almost unique bioreactor, and so we can't really make these broad assumptions, and we really have to be able to understand what's happening in every person's gut to be able to eventually affect and modulate that system. And that's a huge undertaking. Yeah.
Yeah, so this comes down to basically personalized medicine and personalized nutrition, doesn't it?
Yep, definitely. Yep.
Yeah. So I know that there are several groups that are working on this, of course, and I'm just waiting for the future of it, because up to this moment, of course, medicine and nutrition is very generalized, and in the average of all of the different data points that you can have that we are actually getting, but in fact, it actually should be very personalized indeed.
Yeah, definitely. And I think phages and the virome is a great example of that. It is so complex and it is so diverse. It's the most diverse microbial system in the body, your bacteriophages and your viruses. And so while they are the most complex and difficult to understand, I also think they have the most potential in affecting and modulating that system. So there's a lot of potential benefit there if we can sort of crack that microbiome and really understand it.
Wonderful. Okay, let's zoom out a little bit more again into the bacteriophage. Can you say a little bit about how bacteriophages these days are being used, or phages, I guess, in general, are being used in therapy for different conditions?
Yep, absolutely. So, a really hot topic at the moment is this concept of phage therapy or bacteriophage therapy. And just to put a simple definition on it, what we define as of phage therapy is the use of a bacteriophage or a phage, a bacterial virus, to target and eradicate and remove a bacterial pathogen that's causing an underlying disease in a patient. And phage therapy has a really long and storied history. So phages were discovered over 100 years ago,
Yes.
and the first case of phage therapy was applied over 100 years ago now. And so this has been around for a really long time. In fact, phages were discovered and used therapeutically well before antibiotics. And what happened was when we discovered antibiotics and we started to use them therapeutically, that was such a miracle drug. And I don't think we can really emphasize how important and how much antibiotics have shaped our world today. And when they were first used, it was really a miracle, right? Some of these horrific and very difficult to treat bacterial infections, you would simply take a pill from your doctor, and a week later you'd be better, right? Something that would used to kill you was treated almost overnight. Mm. And so phage therapy, as a result, really died off, and it was very, very fringe medicine. It was still used in parts of Eastern Europe, and a lot of research was still going on, and phages became more of a fundamental scientific, almost academic research topic. But all that changed 10, 15, 20 years ago with the imminent and ongoing rise in antibiotic resistance. And this is a global problem that's is going to affect many people's lives over the coming years and decades. It's getting much worse. And what we're seeing is that particular bacterial infections becoming resistant to all clinically relevant antibiotics that we have. In some cases, we have no drugs that can treat these infections. Right. And so this is where phage research and phage therapy is really coming back. And what was once considered a fringe research, particularly over the last 5 or 10 years, is having a lot of interest in its potential to combat antibiotic-resistant infections.
Wonderful. So the phage therapy that you were talking about before, I mean, how were they actually administering this? I mean, before the advent of antibiotics?
Yeah, so I think one of the amazing things about phages is that you can really administer them in a whole plethora of different ways. They're very stable. They, your body is full of phages, so adding more phages to your body, for the most part, doesn't seem to have any adverse effects. So they're quite safe. They have a very high safety profile. So you can drink phages, you can inhale phages, you can inject phages, you can apply them topically on a wound. I would say the important aspect of phage therapy is getting the phage to the bacterial infection. So wherever that bacterial infection is, the best way that we can administer the phages to allow the phages to recognize, infect, and replicate within them is generally the best way to treat that infection.
And that's interesting because if phages are very, very specific to their hosts, then there is no real sort of side effects in terms of, you don't have to worry about it attacking any other type of cell, essentially.
Yep, exactly.
So there's huge specificity there.
There's huge specificity, and it comes back to that microbiome point, right? Antibiotics, are a wonder drug, and when we need them, they are the most important thing that we have in our toolkit. But there's a lot of side effects, right? Antibiotics broadly kill bacteria, and they have huge effects on your microbiome, and that can have long-term, lifelong effects on people's metabolism and immunity and a whole range of things that we're just beginning to understand. So in that respect, phages have a lot of benefits. They have no side effects. They do not infect or kill human cells, but they're also there's a lot of limitations, and it's difficult to use them currently.
Yeah. Okay. Well, thank you very much for that. Is there one last thing, perhaps something that is, a very cool sort of experiment or a cool result that you have that you want to talk about?
Yeah, so maybe I can talk about some work that we've just sort of about to publish. It's sort of been some work that we've been working on for 3 years or so now, and it's been sort of working in the gut environment. So we're working on this gut on chip system where we can build a human gut on a microscope slide. And what we've been doing here is we've been asking the question: can a bacteriophage evolve not to better infect a bacterial host, but evolve to better persist and interact in the human gut? So asking the question, can we get a bacteriophage to adapt to stick and persist in your gut environment, and by doing so, giving additional benefits to your microbiome and that top-down control. And so what we found, just really, really briefly, is that we have been able to show that a particular phage that we're working on is able to accumulate mutations to better persist and adapt within the human gut environment. And it does so by mutating a protein that allows it to stick and adhere to the mucosal surface in the gut. And so we're seeing that this mutant phage had better capacity to persist in that mucus layer, and therefore better capacity to replicate on its bacterial host. So that's been some really cool work, and it's sort of pushing the boundary of what we understand the virome as, right? The virome's always been this entity that predates on the bacterial host, but we hope this work really shows that, well, the bacterial host is not there in isolation, right? There's a huge influence on the human host, on that ecosystem, and just the same way that the phage will drive and target towards this bacterial host, it also recognizes that human and may go along the ways to sort of answering why is our virome so stable, and how can the virome persist within a single person over their lifetime. And it's interesting to consider that these phages are likely evolving and adapting directly towards you and your body.
Wonderful. So, one last question. Obviously, you're
Sure.
doing this in a organ on a chip sort of system.
Yeah.
How do you test this in an actual sort of animal, in an actual sort of animal model?
Yeah. That's a big question. That's sort of a really,
Yeah.
really open-ended question. And I guess, the sort of research that my lab does is quite reductionist in that respect. We really try to understand mechanistically how something happens, and trying to build an experimental model system that we can test this in lab and figure out how this works. And then hopefully we can eventually move that into more and more complex systems. The ultimate goal is to do this obviously in the human gut, or in the real gut environment.
Sure.
And we hope to get there one day. I would say it's just the complexity is too huge at the moment. But by understanding in a really simplified reductionist system how these systems work, and how the phage, the bacterial host, and the mammalian host all interact, we can start to unravel and think about how this happens in the actual gut in such a complex ecosystem.
Wonderful. Great. Thank you so much for your time, Jeremy. This has been so interesting. And it creates a lot of hope, right? Bacteriophages as a way of therapy, bacteriophages as a way of modulating the gut microbiome, perhaps, even, curing curing conditions that have plagued human beings for a long time. So what an exciting area of research this is.
Absolutely, no, it's an amazing field, and I'll maybe give a shout out to any other sort of biologists who are looking for a research topic. I can't recommend phage biology more. There's so much to discover, so much excitement. It's a really growing field. So, but thank you for having me. It's been really exciting.
Perfect. Thank you so much for your time. And this is again We Talk Science. Thank you very much for listening. And till the next time.